Hello everyone, welcome back to one more video session. In this session, I'll be dealing with Mendel's dihybrid cross experiment. It is also called as two-gene inheritance. That means actually we have studied about law of dominance and law of segregation, which are applicable for both monohybrid as well as dihybrid cross. Now, Mendel, after conducting this experiment, he got a different results. Based on it, he proposed one more or formulated one more law or general rule actually, that is the law of independent assortment. First we see the experiment and after that let's see understand the law of independent assortment. The meaning of two-gene inheritance here, in the sense Mendel actually selected two pure line plants for his cross. Each plant actually, in each plant he considered two different traits. Okay, what is that trait? He considered one plant as dominant trait, that is the plant contained a round shaped seed with yellow color, which is crossed with wrinkled seeds with green color. Here wrinkled and green both are actually recessive character for round and yellow color. Okay, so that's it. It's very simple. Let us see the experiment. Okay, so here what I'm going to do is first I'll write the possible alleles. Capital R is responsible for round shape. Okay, it is for round shape. No need to write in the examination, just for your understanding. If it is smaller, that is recessive, then actually we write wrinkled shape. Okay. If I write capital Y, which is exactly opposite, that is the yellow color actually. Okay. And here if I write small y, that is actually the recessive character for the yellow color, that is green. Okay, so that is the green color. Okay, wrinkled shape. Fine. So this is dominant over this is dominant over this. So Mendel considered round and yellow in one plant and wrinkled and green in another plant, and he crossed it. Let us see the cross. Okay, here we write homozygous. So I write P1, that is the parental generation 1, homozygous round. So this is the genotype for round yellow plant, round seed, yellow color. Okay, so I'll just write, I use yellow color. So see the shape, this is round seeds with yellow color. Okay, that is one plant. It could be, you can take it as the male or female plant, nothing else actually, you can take whatever. Okay, here we write homozygous. Homozygous, small r and small y, that is a wrinkled green, a seed. We just use the green color, and this is actually the wrinkled seed with green color, which is recessive. This is dominant. That's it. Both are homozygous, both are pure line plants actually. Okay, so let us take the gametes now. So I'll write G1. The possible gamete, you know that from R you can take only one R actually, that will go into one gamete, and Y in that only one Y will go to the same gamete. So that means R and Y. So this gene is different, this gene is different for a particular character. Correct? There are two characters. Okay, not all four genes will enter into one gamete. You know that gamete contains only haploid, that is actually the chromosomes or genes. Okay, so now again what we do is same, another gamete of this particular plant, the recessive plant, that is smaller small y. Okay, during the fusion of the gametes, what you get is you get R, small r, capital Y, small y, which is heterozygous. Please mention this. I have observed many students don't write the F1 genotype. You don't write it. Please write this. It is heterozygous. If you don't write it, you are going to lose half mark. Okay, heterozygous. But the phenotype is round yellow. Heterozygous round yellow. Okay, so genotype is entirely different actually. It is not like this parent or this parent. It is entirely different. Got it? Round yellow. Okay, we just write F1. Okay. So now we have to self this same like Mendel's monohybrid cross experiment. So we write P2. The P2 here we are going to self it. Okay, self the F1, that is self pollination. In the bracket, please mention self it. Okay, so before you self it, obviously you must take out the gametes, the possible gametes. So this is a dihybrid cross. Here you get 4 different gametes. Okay, so gamete number 2 here. See here, I am taking capital R in one gamete. and then this one, the capital Y in one gamete. Very simple to take the gametes. Many students get confused with taking the gametes. Okay, very simple actually. First you take capital R, then Y, then again capital R, small y in another gamete. Capital R, small y. Okay, again you consider this small r with capital Y. So small r, capital Y. Again small r, small y. Small r, small y. That's it. And now from this parent we can take the gamete that is same. Again I am considering like that, capital R, capital Y. Again capital R, small y. Okay, then small r, small y. Small r, capital Y, sorry. And again small r, small y. Okay, that's it. Now actually we have to cross, like let us see the fusion of these possibility of gametes fusion. Okay, as a result what kind of genotypes we get it. So I'm going to erase this, please copy it. Okay, I'll just draw the Punnett square. Okay, now we are going to draw the Punnett square. Okay, so we need 16 small boxes because we are going to get 16 different offsprings. Let us see that. Okay, so now actually 16. Okay, small squares are there. Let us consider this side as male gametes and this side is for that is actually the top of the Punnett square is for female gametes. Let us write the gametes here, that is male gametes. Again I am going to write this. When you get this question, take your time. Okay, don't hurry. It may actually leads to a different answer actually. Okay, it leads to a lot of confusion. Even though you have studied this concept particularly in your 10th standard, don't be overconfident. If a twisted question is asked particularly for NEET examination, you'll be in trouble. Okay, so take your time, no issues. Okay, then okay, simple. Now actually you have to cross. Okay, for possibility number 1. So what if this gamete fuses with this gamete of the female? Okay, we get capital R, capital R, capital Y, capital Y. Again, another possibility, that is capital R, capital Y, small y. Okay, again, capital R, small r, capital Y, capital Y. Again, capital R, capital Y, small y. This is the possibility. Now we got 4 offsprings with different genotypes. Okay, again. I just want to tell you one thing, whether it is a monohybrid cross or dihybrid cross or trihybrid cross or tetrahybrid cross, doesn't matter. Whatever the number of genotypes you get or offsprings you get within this Punnett square, remember that it is the recessive genotype, which is like a recessive parent, will be always 1. Whatever it is. Okay, if you consider monohybrid cross, out of 4 offsprings we get, that is recessive genotype will be 1 out of 4. Okay, in the case of like we got 16 offsprings, in that recessive one is only 1 out of 16. If you take the trihybrid cross, in that you get around 64 offsprings, you get a huge number of offsprings, that is 64. Out of 64, again there is only 1 offspring which is recessive, like the recessive parent. Got it? 1 by 64. That's it. No need of confusion. There is no confusion at all. Okay. So now we write according to the genotype, we write the possibility of phenotype here, that is round yellow. Okay, so we have one round yellow here and again round yellow. Again, this is also round yellow. Write the phenotype here. I am going to write the phenotypic ratio. So phenotypic ratio, very very important. Don't miss this. You lose. If you don't write it, you lose half mark. Be careful. How many round yellows are there? 1, 2, 3, 4, 4, 5, 6, 7, 8, 9. So we have 9 round yellow. So below that I just write round yellow. Okay, is to let us consider like how many are round green. So round green, 1, 2, 3. So we have 3 round green, 3 I'll just write here. So 9 round yellow, 3 round green is to, okay, so we consider and one more character, that is wrinkled yellow, 1, 2, 3. Okay, so 3 wrinkled yellow, and finally we know that only one wrinkled green. So this is the phenotypic ratio actually, that external appearance of that particular seed, that is actually 9:3:3:1. Remember, very very important. What is the phenotypic ratio of monohybrid cross? Okay, 3:1. Okay, so now genotypic ratio, I need space here, so I'll write here genotypic ratio. Okay, if I go on doing it, obviously it will take a lot of time actually. I know the genotypic ratio, I'll straight away write it because you can easily observe the genotype actually. Capital R, capital R, capital Y, capital Y. How many are there? So I think only one, I guess, correct? So we write like 1:2. Then actually if you take round yellow, then again particularly this particular genotype, okay, find it out. There are two actually, I know it. So I just keep on writing it. 1:2, 2:2, 2:2. 4:1:2, 2:1:2, 2:1. That's it actually, you can do it actually. No need of writing all these things. Instead of that, please remember this 1:2:2, 2:4:1:2, 1:2:1:2. Okay, so here if you just count it, if you look into the phenotypes, 9 plus 3 actually 12 plus 3 15 plus 1 16. So total 16 offsprings. Here also 1 plus 2, 3, 3, 5, 9, 10, 11, 12, 13, 14, 15, 16. So total 16 offsprings, correct? So that's it actually, that's all about the dihybrid cross. Now let us see the test cross of the dihybrid cross experiment. Now let us see the genotypic ratio for the test cross for particular this experiment, that is 2 gene inheritance or dihybrid cross. So we will write test cross. Always remember, in the examination you will get a question like what is the ratio for test cross. The ratio in the sense like we have to determine the genotypic ratio of the test cross. It's very simple. In monohybrid cross we got 1:1 as the test cross ratio. Okay, so let us see for dihybrid cross. Here we know that I'm not going to take a homozygous red round yellow plant. Okay, so I'm going to take its heterozygous plant, that is small capital R small r, capital Y small y, easy, because we know the logic behind it. So that's what I'm taking a dominant phenotype. Okay, so which is crossed with its recessive parent. We know the genotype of the recessive parent. Okay, so possible gametes same, correct, and finally smaller small y. So this is the possible gametes, and for this there is only one possible gamete because everything is recessive. Small r and small y will be present in one particular gamete. That's it. So now the cross is very simple. Okay, we put 4 boxes because we know that there is only one gamete in the female side. Okay, so here we write the gametes of this one. Okay, smaller capital Y and smaller small y. That's it. And here we write that's it. Okay, so the cross, correct. Okay, then finally R, then small y. Here we get smaller capital Y, small y. Here we get smaller small y. If you look into the genotype, all genotypes are entirely different. Out of 4 offsprings, all offsprings having different genotype. So the ratio is, so the test cross for dihybrid cross ratio is 1:1:1:1. So this is actually the genotype. Remember, for monohybrid cross it is only 1:1. Whereas for dihybrid cross, the test cross ratio is this. Remember, very important. It could be asked in your NEET. Okay, so this is all about the test cross. Now I'm going to talk about the Mendel's law of independent assortment. After seeing the result, what he got in the dihybrid cross experiment, he formulated a general rule called as law of independent assortment. I'll erase this. After the completion of the dihybrid cross experiment, that is 2 gene inheritance, Mendel was surprised to get 2 different new phenotypes in the F2 generation. What are they? Okay, we'll write here, that is wrinkled yellow seeded plant, wrinkled yellow seeded plant, wrinkled yellow seeded plant. Okay, how many we got? 3 plants were wrinkled yellow in F2 generation after the cross between heterozygous round yellow plants. Okay, and then we got round green seeded plants. How many? 3. Round and green. Okay, round green seeded plants in the F2 generation, which were 3 actually. Correct. So it was surprising for the inventor. Like, what made them to be like wrinkled yellow and round green? Because these characters are completely absent in the parental type. Correct. So he crossed round yellow seeded plant with wrinkled green seeded plant actually. But it's different, entirely different actually. Okay, 3 and 3. Okay. So after that he formulated the law of independent assortment. It's very simple. When two pairs of traits are combined in a hybrid. Okay, what is the two traits? R, Y, Y. See here, these are actually the two traits. One is for round and another is for yellow, which is actually crossed with smaller, smaller, small y, small y. This is actually the wrinkled character. This is actually the green color character. Correct. Which are now combined in a hybrid. Yes. What is that hybrid? The F1. We got heterozygous round yellow. That is actually the hybrid. Okay. So now when we self it. Okay, so segregation of one pair of character, I'm talking about one pair of character, that is round or yellow. Okay, segregation of round or segregation of yellow. Okay, so one pair of characters is independent. They are obviously independent. Now you can recall the law of segregation, like I said about the gametes. Obviously they segregate independently during the formation of the gamete. Correct. Same here. Actually the characters also segregate independently. Okay, characters is independent to the other pair of characters. So that means round and yellow always never occur in a seed or a plant actually. It can actually separate from each other. That is the reason why we got a different result in the F2 generation. That is 3:9, 9:3:3:1. Here actually these two trees are wrinkled yellow and round green seeded plants. Okay, it's a new generation, okay, or new phenotype we got in F2 generation. So that's all about it actually, it's very simple. Okay, so